Hello and welcome to my YouTube channel. Today I'm going to discuss about gram staining. Particularly today I will be talking about the procedure of gram staining, how gram stain perform in microbiology lab. Today I'm gonna discuss about it. So let's start gram staining. So gram staining is a differential stain which distinguish between the group of bacteria, that is gram positive and gram negative. It is based on their staining ability. Both the bacteria have different ability to stain. Therefore, the name is differential stain. So, in 1884, Hans Christian Gram, while studying origin and causes of pneumonia, developed a stain which is called gram staining. So, gram word is from Christian Gram, which is a unique to bacterial cells due to their cell wall construction. So first of all, for the procedure, what we need to do is, firstly, we have to prepare a smear and fixed by gentle heating. Then primary stain, that is crystal violet, is added for 1 minute. After that, uh, it should be washed under running tap water. Then we have to add mordant, which is a gram iodine, for 1 minute. After that, again wash under running tap water. The slide or the smear is decolorized with acetone alcohol for 10 to 15 seconds. In this step, what we get is actually gram positive bacteria retain the color of primary dye while gram negative bacteria washed away. After that, wash under running tap water again. And the last step is secondary or counter stain with safranin for 30 seconds, which gives color to the gram negative bacteria. Here is the result. Gram positive bacteria get colored by crystal violet, while gram negative bacteria stained with safranin. So gram positive bacteria will be appeared purple, while gram negative bacteria, because they only stain with safranin and therefore they are pink in color. This is the procedure of gram staining. Thank you. In another video, I will be discussing about the cell wall construction of both gram positive as well as gram negative bacteria. Till then, thank you.